Okay, so yeah, it's super great to be here with the Eterna community. It's awesome to see people's names that I recognized from the paper and from all the ASO names that we created. So based on your designs, and so my goal for today is to give sort of an overview of our project and show you some of the results that you may not have seen, and then uh, tell you a little bit about maybe future puzzles that we're thinking about. So I think the exciting bit is that there is a manuscript that's ready for submission and it's been shared with the community. And I think you can still find access to that maybe in the forum. But yeah, but I think it's going to be it's just good to show you guys what's going on. So and again, Victor couldn't be here, and so I've been actually in a deep grant writing frenzy with my colleague Michael Stone, and so Victor sent me these slides last night, so I'm just going to wing it. So we'll see how we can go. So one of the things that I think is really remarkable is this notion of rare disease and what a massive challenge it is to the healthcare community and to the industry. And that's because although these diseases typically are exceptionally rare in terms of their individual prevalence in the population, so typically classified as 1 in 5 to 10,000 live births. Um, if you sum all of these rare diseases that are out there, these inherited uh, genetic disorders, there's more than 7,000. And in the US alone, there's more than 30 million patients. The real, the real problem is that there's a massive financial burden associated with treating rare disease. So it's typically much more expensive to treat a rare disease patient. And the real tragedy is that the vast majority of rare diseases have no treatment and no real hope of any treatment. I think the overwhelming um, challenge of rare disease is, when you have a child who's born with a rare disease, it's it can be a grassroots effort to mobilize the scientific community to be able to potentially uh, engage and come up with initially a mechanistic understanding of what a potential mutation is doing, and then the notion of potentially finding a therapy is really a long ways off. And so it's clear that there's a massive um, need to be able to accelerate this process of discovering rare disease therapies and making it much more efficient for clinicians to find their ways to molecular solutions that can help patients. So one of the really the most very promising approach that's if you look at sort of preclinical trials, there's now well over 1,300 different antisense oligonucleotides that are being developed to treat different rare diseases and even some common diseases. Antisense oligos are nucleic acid-based medicines, and they work just as you might expect through hybridization to specific target sequences. These ASOs come in many different formulations, both in terms of their chemical modifications and in terms of um, their mechanisms of action. And so, typically there's really three main or four main ways that that ASOs can work, although there are many new creative ways that people are describing. But typically, you can either make use of um, the cell's ability to take RNA oligos and incorporate it into something called RISC, which can then kill mRNA or block translation of messenger RNA. You can also make antisense oligos that work in conjunction with a cellular enzyme called RNase H, and this can be used to trigger the decay of problematic RNAs. And then in the case of the work that we've done together with Eterna and in our lab, you can generate antisense oligonucleotides that can actually manipulate the process of pre-mRNA splicing, which is a fundamental step in eukaryotic gene expression, and we'll talk about that in a second. What I think is really remarkable is that ASOs are just like the mRNA vaccines. It's not a new technology. It's something that people have been studying for decades, and only recently has you know have ASOs really become FDA approved and useful for treating various diseases. This is just a little timeline of one classical ASO that was used to treat the disease spinal muscular atrophy. It had a pretty similar developmental timeline in that, the work to understand the molecular basis of spinal muscular atrophy began in the early 2000s. And then nearly 15 years later, an FDA approved drug was on the market that could cure SMA. So that's roughly a 10 to 15 year timeline to produce a new drug. And that's sort of typical for most conventional drug development enterprises. But because antisense oligonucleotide chemistry is largely the same, the composition of one ASO is the same as another ASO, just the sequence of those nucleotides is different. Developing subsequent antisense oligos can really greatly be accelerated. So this is an example of a precision therapy that was developed in just 16 months for a patient with an ultra-rare seizure disorder called Batten's disease. And this is a drug that was built for a little girl who came into a clinic in neurology clinic in 2016 with seizures. And then by 2018, the patient had been dosed with a drug that could correct the splicing of that disorder. So I think, that was just, a few years after the development and full approval of Spinraza. We're getting to the point now where when a genetic disorder is discovered, if there is an RNA basis, it appears as though we should be able to quickly develop antisense oligonucleotides that can be effective for ameliorating the disease. So how does this work? So pre-mRNA splicing is a fundamental step in gene expression. I think for those of you that don't know, our genes are pretty complicated. They have lots of different component parts, and only a tiny fraction of that information that's in any given gene, so we have thousands of genes in the genome, only a small piece of it is actually useful for making a functional messenger RNA that can be translated into protein. Those little functional sequences are called exons, right, shown here in this color. And there's these intervening sequences or introns, which shown here in kind of yellow, that separate the protein coding exons from the rest from each other. So our genes are discontinuous. What's interesting is that although this cartoon makes them look roughly equivalent in length, it turns out that, especially in the human genome, introns are absolutely enormous. So 3 or 4 orders of magnitude greater than the exon length. The typical exon is maybe 120, and the introns can be on the order of 10,000 to 100,000 to even 1 million nucleotides in length. So the actual protein coding information is just a tiny little island in this sea of RNA. And so after a gene is transcribed, you make this pre-mRNA that contains both exons and introns. And the pre-mRNA has to be processed in order to generate a competent messenger RNA. And so that one of the key steps is this process of splicing, in which the introns are removed and the exons are ligated. And that generates this messenger RNA that can be translated into protein. So in the case of rare disease, you often have point mutations, either in the exons, quite typically, sometimes in the introns. And most of the time, clinicians have thought that mutations in exons, especially in the protein coding regions, lead to the production of a faulty protein, something that misfolds. Maybe the enzymatic activity doesn't work, maybe the protein's not stable. But more recently, it's been discovered that many mutations can actually cause errors at the step of pre-mRNA splicing. Let me see. So this is an illustration of the crazy mechanism that has to occur in order to splice an intron. Now, in the human genome, there's something like 300,000 introns, and your genes are being, the genes that are active, they can be transcribed many, many times in the life of a cell. And so every time you make an intron, you have to assemble this huge complex. So here's a pre-mRNA up here. And as the intron is sort of synthesized by RNA polymerase, many, many proteins and RNAs have to bind to this intron in order to create a complex called the spliceosome, which then can catalyze this organic chemistry of chopping the intron out and ligating the exons together. And it's really not super critical that you understand all the different steps here, but people have been studying this for decades. Everything that we're going to focus on happens really early in the splicing pathway, where some key regulatory proteins and core components of the spliceosome bind to the intron and bind to some of the exon sequences and help to promote the earliest step of spliceosome assembly. And this is the step where the identity of the exon is established. So you can imagine that these tiny little bits of exon, on occasion, if the spliceosome gets it wrong, you could omit some of that sequence. And if I just go back one slide, a real issue here is when you omit some of this useful protein coding information, you either get a faulty message or you get a protein that doesn't do its job. And that's what underlies many different inherited diseases. Okay, so this slide just shows you some of the anatomy of a typical intron. So we're looking at a pre-mRNA from the perspective of the intron. The exons are shown in gray, and this is the part that we're most interested in getting spliced into the messenger RNA. There are three really critical regions of an intron. And the top line here is a very simple eukaryote called S. cerevisiae, or brewer's yeast. And the size of the nucleotide sequence here, this is sort of a shows you the probability of finding this particular base at this particular region of the intron. So the very beginning of an intron is defined by this GU at the 5' end. And in yeast, you can see many more of these positions are pretty well conserved at the 5' end. In humans, it's much more degenerate. So you still have this strong preference for GU here, but the rest of it sort of falls away and becomes less complex in terms of, or sorry, more complex in terms of its sequence composition. This region is called the 5' splice site, and it's a key landmark in sort of describing the boundary between an exon and the start of an intron. Another element is this branch point sequence, and this is important because you have an adenosine here, shown here in this TAAC, that's involved in the actual catalytic step of splicing. And so one of these branch point adenosines has to actually perform a nucleophilic attack at the 5' splice site. In yeast, that sequence is well conserved. It has this TAC or UAC, UAAC, or WAC is what we call it, sequence. But in humans and other metazoans, multicellular organisms, this sequence is highly degenerate, and it's very difficult to even predict computationally where a branch point is. And the last key sequence element is called the 3' splice site. And this defines the 3' end of the intron. And the main feature that you can see here is that you've got a conserved AG dinucleotide at the 3' end in both yeast and in humans. And then you've got really a little bit less of a preference here, but there's a kind of a pyrimidine-rich region in the yeast. In the humans or metazoans, you have a much stronger preference for a polypyrimidine sequence near this AG sequence. So the big take home from here is that when you look at splice site sequences between humans and yeast, in yeast, you have very short introns with really well-defined splice sites. In humans, you have really long introns with poorly defined splice sites. And so the question is, how does the spliceosome, which basically does the same job in both systems, find and assemble on introns when they have this very degenerate, very difficult landscape in humans? And it seems like we get it right most of the time. And part of the solution is that there's a number of other sequences that help to specify where the splice sites are found. So you can imagine that although the splice sites themselves are more degenerate, there's auxiliary sequences, so we call them splicing silencers and splicing enhancers, that can help to specify where the correct splice site is. And so in this slide here, I'm showing you kind of a 3 exon gene with 2 introns, and I've highlighted just a couple of examples of potential auxiliary elements. So over here, we might have in red a splice site silencer near this 5' splice site. Down here, we might have an exonic splicing enhancer that's near this 3' splice site. This might promote splicing. Down here, there might be a splicing silencer that could repress splicing, an intronic splicing enhancer that could promote splicing, and so on. And so one of the keys to splicing in multicellular organisms and humans in particular is that you have this combinatorial set of interactions between these auxiliary regulatory elements and then these core splicing signals called the splice sites. And the auxiliary elements play an important role in specifying or helping the assembling spliceosome figure out where to do its job. So this is a zoom in now on the exon level, right? And so in this case, what you're looking at is an exon. And now instead of just focusing on the cis elements, so these are the splicing enhancer shown here in green with this GAA motif. Here's the 3' splice site and the polypyrimidine track. I'm also showing you some of the transacting factors that can interact with these sequences. And this sort of cartoonish looking set of proteins over here are components of the spliceosome called U2AF, U2 auxiliary factor, and U2 SNRP, which is a key component of the spliceosome that recognizes that branch point. And down here we have the U1 SNRP, which is another sort of key component of the spliceosome that binds early in assembly. And these enhancers tend to be recognized by RNA binding proteins. And in this case, this guy is called an SR protein. It has a serine and arginine rich domain. And then it can interact with the RNA through specific RNA recognition motifs or RRMs. These proteins work to, we don't really know how they work, to be honest with you. We know that they bind to the exon, and we know that somehow the exons splice better. It's not really clear if there's an RNA structural rearrangement that occurs, or if it's simply that this domain called the RS domain can interact with components of the splicing factors directly. That's one of the hypotheses. I think the work that we've done with Michael Stone and Riju and the Eterna community suggests that RNA structure is going to be an important part of how enhancers and silencers work. Anyways, so you have these positive acting proteins that can recruit. Sorry, that's my dog. I gotta let him out. Hold on. So you've got these positive acting proteins that can recruit components of the spliceosome. These can be opposed by splicing silencer proteins, or the HNRMPs, that want to either repress the activity of the SR proteins or somehow block the function of the SNRPs. And what's interesting is that inherited disease-causing mutations can rewrite this regulatory code so that now a sequence that was benign might recruit one of these repressor proteins that can then lead to this exon appearing to be an intron sequence and be ignored by the spliceosome. And this happens, there's many different ways that this happens. This sort of illustration here lists a few ways that human inherited disease-causing mutations have been shown to corrupt the splicing process. So just going from left to right, mutations have been identified at branch point sequences that block intron exclusion, mutations at both the 3' splice site and the 5' splice site. And so this is essentially changes at that GU sequence or the AG sequence. They can absolutely block splicing. And it's been shown by other groups that 10% of all inherited disease mutations work by corrupting that signal. It's also clear that you can have mutations that create exonic splicing silencers. And so this has been demonstrated for the spinal muscular atrophy disease gene, SMN2. You can have mutations that cause the loss of splicing enhancers. And so you lose the recruitment of these positive acting proteins. And then you can also have mutations that function by actually working in an opposite direction, in that you can convince the spliceosome that there's a chunk of the intron that could possibly be spliced into a message. And so now you've put some garbage sequence into a messenger RNA and totally corrupted it. Often that leads to the messenger RNA getting degraded, but sometimes you make totally faulty protein. And so what happens here is that sequences that look like exons, we call them cryptic exons, can be activated by mutations. And this is the example in that Batten's disease, which is that seizure disorder that I mentioned earlier. And so here you could imagine that masking a potentially cryptic splice site could be a good way to rescue the function of that gene. So a classic example of sort of inherited disease that has a a splicing phenotype is spinal muscular atrophy. And this is really the first FDA-approved ASO for rare disease that fixes splicing. And this is the work from Adrian Craner in particular at Cold Spring Harbor. And I just thought I'd show you this because, well, it's just such a great example. It's also highly impactful because this is the leading cause of infant lethality. So 1 in 10,000 children are born with this disease, and in most cases they won't make it past their six-month sort of birthday. This is a typical spinal muscular atrophy is a typically rare inherited disease, which means that it's going to be follow classic Mendelian genetics that's recessive, meaning that if you have two parents, both of which are carriers of the mutated gene, you can actually have a 1 in 4 probability of getting a recessive child that's born that has the disease phenotype. The molecular genetics of this disease are really interesting in that this gene SMN1 and SMN2, it's on chromosome 5, and essentially you have 2 copies on 1 chromosome for this gene. SMN1 and SMN2 are called paralogs, so basically they have the exact same sequence. They, in theory, could do the same job, but in practice, it turns out that only SMN1 produces functional protein, okay? And what's interesting about SMN1 is that this gene has got 8 exons. All 8 get spliced efficiently from the SMN1 locus and make lots of SMN1 protein, and everything's great. Your SMN2, oops, what did I just do there? Your SMN2 gene, on the other hand, has a single C to T mutation in exon 7. And this is actually a totally silent synonymous mutation. So it means it doesn't change the underlying genetic code. If you were to use the computer to translate the messenger RNA from SMN1 and SMN2, it would make the same protein. The interesting bit here is that this C to T mutation, which is encoded in every copy of the SMN2 gene, so we'll call it a polymorphism rather than a mutation. This it corrupts the splicing regulatory information in this exon and causes this exon to look like an intron. And so the vast majority of messenger RNA that's produced from SMN2 skips exon 7 and makes an unstable, unhappy protein. So you get very little functional protein out of this gene. And in individuals with spinal muscular atrophy, it turns out that this SMN1 gene is usually deleted. And so you're essentially running your cells on a single copy of the SMN2 gene. What can happen in some patients that have sort of less aggressive forms of disease, that you'll actually get extra copies of the SMN2 gene, and that'll sort of attenuate the phenotype a little bit. But the underlying molecular basis is that this C to T change corrupts splicing of SMN2, leading to production of a faulty message. Over the course of, I would say, that 15-year period, Craner's lab and Jim Manley's lab worked out the mechanism of how this exon 7 is recognized by the spliceosome and why it goes wrong when it has this C to T mutation. And at some point, these two labs went from arguing about the mechanism to Craner's lab actually deciding, well, okay, even if we understand the mechanism, it's really important that we actually go and take action and try to rescue this gene. And so they shifted gears and began to develop antisense oligonucleotides that could rescue splicing. And they used essentially short oligonucleotides that look a lot like RNA, except for the fact that there's some chemical modifications, such as this thiol group on this phosphate that links the sugar phosphate backbone of the oligonucleotide, as well as this 2' hydroxyl, which instead of having a methyl group here, actually has a methoxyethyl group. So typically we have a hydroxyl. Some oligos will have a 2' O-methyl group. Craner's lab uses 2' MOEs. And the idea here is that these modifications improve the stability of the oligo in the cells. They improve the specificity and the affinity of the oligo for its target. And then they also, in some cases, with the 2' MOE, can reduce potential cytotoxicity. So these are really key modifications that help to make it, a nucleic acid into a suitable therapeutic modality. So the way this antisense oligo works is that it actually binds to a site in the intron downstream of exon 7, which prevents the recognition of repressor proteins that then silence this 5' splice site. So if you include this oligo, you can actually rescue splicing of exon 7 and then make full-length SMN2 protein. And what's very clear is that the earlier that you recognize that you have a child that's been, is going to be expressing 2 copies of the mutated gene, the sooner you can give them the antisense oligo, the better outcome they have. And so prenatal screening for SMN2 is now becoming a pretty general approach across the world. That's leading to earlier and earlier detection of spinal muscular atrophy and then better patient outcomes. So my lab has been studying splicing for a long time, and many years ago, I guess 2011, we published a big study of genetic variants, human inherited disease variants that we thought could affect splicing. And so I guess coming from the SMA work, the idea was, how many other diseases are out there that could have splicing as their underlying etiology. And in this survey of thousands of inherited disease mutations, we found that roughly a quarter of missense and synonymous mutations could be predicted to impact splicing. And there's a pretty big and long list of genes that we came up with that had a high prevalence of splicing, but sort of potential defects. In the paper, we only tested a couple, but we left thousands and thousands of variants untested, and many of these genes are related to diseases including cancer and clotting disorders and kind of you name it. It's across the full spectrum of the rare disease space. And so we had many hypotheses that were untested. We turned our attention initially to this F8 gene in a follow-up study that Victor's talked about a few times. And we focused on this guy because it had sort of the highest proportion of potential variants that could affect splicing. And so this made it very easy for me to recruit a large-scale undergraduate research lab and get them to essentially in parallel test many different mutations in the F8 gene. And so they did this using a reporter assay. This is the bread and butter of what we call the toxic RNA lab. The idea is that we make this Frankenstein sort of molecule that has basically the human beta globin locus. So this is encodes the beta globin gene. It has 3 exons, 2 introns. And these introns are relatively small. This whole locus is 1,500 base pairs. And so this means that you can put it into a plasmid and actually manipulate it in the lab, create mutations, et cetera. And it's a great system for us to take what we think are going to be fragile exons that can be misspliced when they're carrying an inherited disease mutation, embed this exon and some of the flanking intron sequence into this beta globin gene, then ask how exon 1 is spliced to our test exon and then spliced to exon 2 and 3. So this gives us a model system that lets us survey the effects of different disease-causing mutations on a wide array of human diseases. And so in this case, I know Victor has shown you some of this. We tested, and this is again undergraduate students in the lab, tested 97 different hemophilia A causing mutations in the F8 gene. They looked at 11 different exons. And what was really remarkable is that the vast majority of exons were really refractory to aberrant splicing in this context. That is that most of the predictions that we made didn't have an impact on splicing. However, in 4 different exons, and this one in particular over here, they appeared to be highly sensitive to aberrant splicing. And so this is exon 16 from F8. And you can see here, if you look at our wild type F8 exon 16, which has been included in the beta globin reporter, you get a single band from this splicing reaction. This is plasmids transfected into cells, RNAs extracted, and then analyzed by RT-PCR. the singular band corresponds to the included form of this messenger RNA, which has beta globin sequences plus the F8 exon spliced in. But you can see as we introduce these different point mutations, there's an increase in exon skipping. And several different positions in the exon have strong effects on splicing. In particular, the site down here, C5543A to G, has a really strong impact on exon inclusion. And this is simply a graph of these data here. This is a representative gel. This is summing many different biological replicates. Percent spliced in is simply a measurement of how many molecules have this exon included versus the total number of spliced messengers. So exon included over the included plus skipped. And so then you can put a number to how big an impact a mutation has on splicing. So I'll skip that. So I don't have time to go through the whole paper, but the upshot is that we solved many different RNA structures using shape in order to determine how different mutations were impacting splicing. And while we generally saw in blue here, you can see these beautiful arc diagrams of the wild type exon 16, and in red here is our favorite mutation, that A to G mutation at the end of the exon. You can see that there's some subtle differences in pairing in the exon. We were really drawn to this sequence here that had this interesting looking three-way junction structure. And what we noted about this structure is that many of those key sequence elements, so the branch point and the polypyrimidine track, are actually embedded in this secondary structure. We also noted that the 5' and 3' splice sites in all of the exons that molecules that we looked at are in close proximity to each other. And so this is an extensively structured exon that has its polypyrimidine track, 3' splice site, and branch point embedded in a secondary structure. We were also interested in identifying antisense oligos that could rescue splicing. And I'm not going to show you those data either, but the key take home is that we found antisense oligos, and it turns out that oligos that target the structure can rescue splicing. And so we're working on understanding mechanistically what's going on there. But what we discovered in doing these experiments is
Jeremy,
that,
can I put in? Victor and I didn't tell you that he only signed up for a 30-minute slot. So
how many minutes are we at?
We're at 30. Yeah.
Oh,
Which
well,
is fine. Can you skip? I mean, a lot of us have seen
yeah,
this.
I'm right at the end anyways, so.
Okay, cool. Okay, thanks.
So anyways, the important thing is we built this whole thing is expensive and cumbersome and time consuming. And so we thought maybe there's a better way. And so we engaged you guys, created the OpenASO Rescue Challenge. And the idea here was if the players could find a better solution than we did in terms of finding something that could alter the structure of the pre-mRNA, and then we would test those in the lab. And this process yielded a bunch of different compounds, and so that's all shown here. I think Victor had shown you this slide here, and what was interesting is that the Eterna players basically found very similar solutions that we did. And so you can see we're looking for molecules that allow us to go from mainly skipped exons to mainly included. And so these three here, strong impacts on splicing. These guys over here, very strong impacts. And so, in fact, some of these ASOs worked better than any of the initial ones that we found in the first place. In our paper, we found that a cocktail of oligos that could rescued splicing almost completely by binding to this entire sequence here and liberating the branch point in the polypyrimidine track. That was a very effective drug. And we found that combining different combinations of the Eterna lab players, ASO designs actually also resulted in almost complete rescue of splicing, almost as well as our previous design. So yeah, the last bit is just that, what we've done, I think, is that in this work, we found a really cool RNA structure function relationship for fragile exons. I think we've demonstrated with Eterna that we can come up with solutions for rare disease splicing sort of from first principles using RNA structure, and that we're currently in the process of creating new challenges for the Eterna team, focusing on a variety of different inherited disease genes, including BRCA1, galactositis alpha, which causes a pretty horrible disease called Fabry syndrome, and MSH1. These are all genes that are involved in rare diseases ranging from cancer to Fabry, and we found fragile exons in each of these cases. And again, we're interested in finding ASOs, but we'd like to do it more effectively, and we think that your group can help us do that. So, like I said, Victor has just graduated. He's going to a new lab. We'll continue doing this work in collaboration with Michael Stone's lab, obviously continuing to work with the Eterna team. And these are the people who put Victor through his paces. And we've been supported by funding from the National Institutes of Health and the University of California, Santa Cruz, and some other funding, including the NSF. So yeah, thanks a lot. And sorry to scare you guys. I wasn't going to keep going, but that was
yeah, I want to make sure you were aware. So Harry Noller was on his exam committee, huh? That's fun.
Yeah, he had Harry and Carol Greider, so a breakthrough prize winner and a Nobel Prize winner. So that was
yeah. Can you go back to the cocktail slide? That's the one I really wanted to highlight to people.
Yeah.
That this is the new data. And your cocktail was 3. Can you highlight where your 3 results, that was your 3 cocktails together?
Yeah, so ours are here. They basically they occlude this entire part of the stem, so that liberates the polypyrimidine tract, the branch point. And then up here in this apical part of the three-way junction, we're pretty sure that there's a binding site for H9RPA1, which is a splicing repressor protein. And so that's one of the key mechanisms. So we think that it's a combination of liberating these two features of the 3' splice site, but then also masking the A1 binding site here. And so the combinations that worked best, it's only a 2 ASO cocktail. They're not contiguous. It's OpenASO 10 and 11. And I sort of forget, yeah, we changed the names for the paper, whose designs those were. But in this case, you have one that's really masking the H9RPA1 binding site, and the other one that seems to be liberating this polypyrimidine track. And this guy basically works just as well as our 3 ASO trio, combination.
Oh, you think so? Okay.
I mean, it's pretty close. They're this would be a non-significant result, I think, if you compare directly between ASO 10 and 11 to the ASO trio. The means are pretty similar, and I think that this gel kind of, when you run a lot of gels, there's only this looks like a very extreme difference. If this maybe had a little bit more sample loaded, you'd still see a little bit of skip signal there. And that's apparent. You can see here, this isn't 100%. It's more like 85. And this is somewhere between probably 65 and 85%. So.
And I have to say, from the clinical side, most likely something, if anything ever did go into clinical trial, they would prefer 2 ASOs rather than 3.
2 for sure. And I think, the interesting bit about the ASO work is that typically once you get a region where there's some kind of signal, like, so for example, this 3-way junction appears to be a good target, you would then go through and do much smaller incremental steps across this region. So we took a very coarse-grained approach because we don't have infinite money. And so we sort of made 18 mers and stepped them across the intron and exon in 18 nucleotide steps. But you can vary the length of these guys, so increasing or decreasing, and then step through it in much finer and finer windows. So even down to a single nucleotide step. And I think with what they call a micro walk across this region, it's likely we could find an ASO that is just a single oligo that actually has a similar impact on splicing.
I think so. Okay, interesting. And explain why to players who maybe don't know, is because when you introduce the ASO into the system, into the body, the RNA into the body, there's a high risk of side effects. And so to minimize side effects, researchers generally want to minimize how many ASOs would be. Usually it's only one ASO so far. I don't know.
Yeah, for sure. And it's just, I think it's yeah, it's a specificity issue. And then the other piece is, yeah, I think in this case, most folks don't have, I think, as strong a structure function relationship as we do here. And so I think it should be possible to destabilize the structure, mask the A1 binding sites all with one molecule.